So I'm pleased to introduce Rose Schrott, who is our next speaker. She is a soon-to-be PhD, currently a PhD candidate at Duke University in the Integrated Toxicology and Environmental Health program. Her dissertation work is focused on the impacts of male cannabis use on the sperm epigenome. She earned a bachelor's degree in biology from Johns Hopkins and a Master of Science in Biochem and Molecular Bio with a concentration in reproductive biology from Johns Hopkins Bloomberg School of Public Health. Rose is passionate about epigenetics and the early life origins of health and disease, and plans to continue to pursue research in this area throughout her career. So with that, I will hand it over to Rose.
Awesome. Thanks so much, Jordan, and thanks everyone for participating in our session today. So the title of my talk is Let's Be Blunt, Paternal Cannabis Smoke Extract Exposure in Rats Induces Heritable Epigenetic Changes in Sperm and Cardiomegaly in His Offspring. So legalized marijuana or cannabis is rapidly expanding across the United States as well as the globe. And with more and more states in the US voting to legalize recreational or medical marijuana programs or even to just decriminalize the drug, we know that access to legalized forms now is really going to be rapidly expanding. And this leaves a lot of questions to be, that remain to be answered, which Dr. Murphy has touched upon earlier today, are what are the impacts of this on the sperm epigenome? And how might these exposures impact not only a man's own health and his own epigenetic profile on his sperm, but his future offspring and subsequent generations? And so again, Dr. Murphy really nicely touched upon all of the human cohort work that our lab has been doing to answer these questions. And today I'll be sharing some of the rodent work that we have been doing in parallel. So some of the initial mammalian model data that looked at paternal or parental THC exposure came from Yasmin Hurd's group, where they used a biparental exposure paradigm, exposing both male and female rats to THC, that main psychoactive component of marijuana or cannabis. Then they mated these animals as adults and looked at offspring for outcomes. And some of the main findings from their work were that they observed increased heroin-seeking activities in the offspring. They observed changes in DNA methylation in offspring nucleus accumbens tissues. And they also observed changes in gene expression of receptors involved in dopaminergic and glutamatergic signaling in offspring. And again, this was all resulting from a biparental exposure. And so our lab has been keenly focused on paternal exposures. And so we wanted to look at this from the paternal exposure side. So we exposed male rats to 4 milligrams per kilograms of THC. Again, this is a dose that reflects about moderate use in humans. And we exposed them prior to conception and then analyzed the sperm of these rats for methylation changes as well as offspring for any heritability. And so here we observed DNA methylation changes in F0 sperm, and we observed heritable effects that altered methylation at select genes, some of which Dr. Murphy presented earlier today as well. Our collaborators observed changes in behavior and learning in offspring born to THC-exposed fathers, as well as deficits in synaptic cholinergic function in offspring born to these THC-exposed dads. And so while, of course, it's important to tease apart the specific contributions of THC because of its psychoactive components in nature, we now wanted to shift to using a cannabis smoke extract model to more closely resemble what humans are actually consuming. So there is cannabidiol in this as well as other combusted extract particles. So in the current work that I'll be presenting today, we used a cannabis smoke extract exposure. We had 3 different groups of rats. The first were our vehicle control rats that were injected subcutaneously with our vehicle control during whatever time period injections were administered. Our second group is what we are calling our early exposure group. So these were animals that received 4 milligrams per kilogram of THC in this cannabis smoke extract for the first 28 days of the exposure, and then had about a 56-day washout period where they either received no injections or a vehicle control to see if we could determine any sort of amelioration of these effects. And then the third group we're calling our late exposure group. So these were animals that received vehicle control initially, and then after a brief period of no injections, they received 4 milligrams per kilogram of THC in the cannabis smoke extract for this latter third portion. After all injections were completed, these male rats were mated with drug-naive females, and then we collected mature motile sperm via the epididymal swim-out method. We extracted DNA and used that for whole genome by sulfide sequencing. So here is our workflow for the whole genome analysis. So similar to how we did with the humans, we first limited this to just our top 10,000 CPG sites, and then we implemented an internal threshold of a greater than 10% methylation difference at these sites. And so this left us with about 3,300 CG sites from the late exposed versus control group and about 2,100 from the early exposed versus control. We asked questions about amelioration, such as are the changes in the late versus control diminished in that early group? And then we used the ingenuity pathway analysis to look at diseases and functions and canonical pathways that were associated with the genes that had differentially methylated CG sites. And then lastly, we used bisulfite pyrosequencing as an independent methodology to validate. So looking first at amelioration, and I'll orient you first to the scatter plot in the middle. So we have the early exposed versus control mean methylation difference plotted on the y-axis, and the x-axis is the late exposed versus control mean methylation difference. And what you can see with the kind of partitioning of all these points into the upper right and bottom left quadrants is that what this tells us is regardless of the timing of the exposure, for the most part, the direction of methylation change was the same across the early and late exposure. Of course, there are some where this shifted across exposure times, but for the most part, methylation differences were occurring in the same direction for the early and late exposure. Now I'll orient you to the bottom left graph, the hypomethylated group, and this is similar to what we observed with our human studies. If you have methylation difference on the y-axis, again, each dot here is the average methylation level at a specific CG site. And so then we're looking here at the late exposed versus control and comparing to the early exposed versus control. And you can see that the magnitude of methylation difference is decreasing in the early exposed versus control versus the late for the majority of these sites. Now moving to the upper right, the hypermethylated group, you basically see the same phenomenon where you have methylation difference on the y-axis again, and each site is that average methylation difference, and you see that the magnitude is decreasing as you move from the late exposed to that early exposed group. So this begins to suggest some sort of amelioration might be occurring. We wanted to look a little bit more closely into this, and so we limited now our analyses to genes that had 5 or more CG sites that were impacted by the exposure in that 10% or greater methylation difference list. And so here, these are genes from our late exposed versus control group that had 5 or more CG sites, and then we looked at those same CG sites in the early exposed versus control. And what you can see, again, really nicely looking at this heat map with the late exposed on the left side of it and the early on the right, is that the magnitude of methylation difference is decreasing. And for the most part, though, the methylation difference is staying in that same direction. There are a few sites where this is not the case, but for the most part, the magnitude is decreasing in the same direction. What was really interesting and kind of surprising to us, though, was when we did that reverse analysis of this. So we took genes with 5 or more CG sites that were significant in that early exposed versus control list, and then we compared them back to the late exposed. We see that there are a subset of genes that have affected CG sites that are more pronounced, that the methylation difference is more pronounced in that early exposed versus control group compared to the late. So there is some sort of temporal specificity appears that is being initiated and then carried through the study time period that we're not totally sure what might be explaining this. Perhaps it's that some of the populations of somatic gonadal stem cells are being affected that are propagating, but only at certain genes. So we're still trying to tease apart why this is happening. But I will say this temporal phenomenon is something we've observed throughout our studies, and I'll present some more data about that shortly. So we used, like I mentioned, the ingenuity pathway analysis to look at canonical pathways. And one thing that I think is always interesting to do is to look at what overlaps. And so we see here that there are overlaps between the late exposed and the early in processes like synaptogenesis signaling, cardiac hypertrophy, and also just many different signaling pathways are coming up with GPCR receptor signaling and other pathways. And then when we look at top diseases and functions, we see that a lot of the terms that are showing up are important for early developmental processes. And again, there is a lot of overlap between these two groups. So we see things like cellular development, cancer, nervous system development, and cardiovascular system development. And we use these terms to kind of guide some of the genes that we then chose to validate with our bisulfite pyrosequencing data. So here for the validation of the whole genome data set, I'll be presenting bisulfite pyrosequencing data. The controls will be black, the early exposed in blue, and the late exposed in green. And I'll go gene by gene for this for a subset of the ones that we've been analyzing. So the first gene here is Pxylp1. To orient you, the top graph here is pyrosequencing data from the 2 CpG sites that correspond to those that were identified on the whole genome bisulfite sequencing data set. You have methylation difference, percent methylation difference on the y-axis, and then those 2 CG sites on the x. And we are looking at this in the control versus a late exposed, because that is what showed up as being significantly affected that we wanted to validate in the whole genome data set. And so, of course, here you can see that we were able to significantly detect and validate a loss of methylation at these 2 sites in the F0 sperm of these animals. In the bottom graph, since with pyrosequencing, we're able to design an assay to encompass a region, we were actually able to capture an additional CG site here. And we also ran this assay with our early exposed group as well. And we were able to detect significant losses across the region, and not only at the late exposed, but also at the early exposed group. So next, moving on to Mtss1l. This was a gene, if you look at the top graph, that actually showed up in our whole genome as being affected in the early exposure group. And so again, methylation difference on the Y and the 2 CG sites we analyzed to validate on the X, and we were able to significantly, to validate a significant loss of methylation at these 2 sites. When we move now to the bottom graph, looking at the entire region that the pyrosequencing assay we designed captured, we saw a significant loss of methylation now across the entire region, but this was one of those interesting genes where it had this temporal effect that was only really occurring or only evident at that early exposure time period, so no significant effects at all for the late. And then the last gene I'll talk about today is GABRB2, a GABAergic receptor. Again, we were able to validate, well, now only one of the 2 CG sites that was identified from the whole genome by sulfite sequencing in the late exposed versus control on the top graph. And then when we moved to look at all of our pyrosequencing data, this sort of data with this kind of temporal dose response, if you want to call it that, is what I would have expected most of our data to look like, where we saw a greater magnitude of difference in the late exposed and a lesser degree of magnitude of difference, but still somewhat in the early exposed. And so it's interesting that we have seen various different patterns when we're analyzing this data. Next, we asked questions about heritability of these altered methylation patterns. So just to reorient you here, the top graph here is going to be that same F0 sperm data. And then the bottom graph is what we chose to validate. And so with PXYLP1, it's expressed in epididymis and specifically in mature spermatids in the epididymis. We wanted to look in F1 sperm. And so we were able to observe a significant loss of methylation present at the late exposed compared to control F1 sperm samples for this gene. For Mtss1l, because it is highly expressed in the hippocampus, we looked in F1 hippocampal tissue. So again, the top graph is the F0 sperm data. The bottom graph now is the F1 hippocampus. And we saw that altered methylation patterns did persist at some of the same CG sites in the offspring hippocampus tissue. But again, we saw this temporal specificity where this was only occurring in the early exposure group. And then lastly, for GABRB2, again, the top graph is the F0 sperm, and the bottom is F1 hippocampus, again, because of the neuronal function of this gene. And we saw a significant loss of methylation that persisted in the early exposed, but only at one of these CG sites. So follow-up work for this is to look at some expression patterns to see if methylation and expression are correlated and if they're being impacted by this paternal exposure. Then the last question we wanted to ask that I'll present here is, does this exposure induce any physical abnormalities in the offspring? And there's a growing body of literature that prenatal exposure to marijuana or cannabis is associated with various teratologies, including cardiovascular deficits and cardiomyopathy. And if you remember back from some of those canonical pathway and diseases and functions terms, we saw things having to do with cardiovascular system development and cardiac hypertrophy showing up as some of the significant terms associated with our data set. And so we very simply just wanted to start off by looking at heart weight. And so I'm representing here heart weight as a percent of body weight on the y-axis. The controls again are in black, the early exposed in blue, and the late in green. And you can see that there is this significant increase in offspring heart weight as a result of the paternal exposure compared to the controls. We separated this out then, looking at males and females separately. In the females, we do see that this increased heart rate is significant for both exposure periods, but in the males, interestingly, we see that this is only significant in that early exposure group. So right now we're working on some staining, looking at proliferative and as well as fat markers to see what might be contributing to this increased heart weight phenotype that we have observed. So with that, some conclusions. We have observed that cannabis smoke extract exposure induces DNA methylation changes in sperm. We have initial evidence suggesting these methylation changes are heritable at select genes. And we observed trends that suggest amelioration of the effects of CHC exposure following a washout period at select genes. We observed heart size abnormalities that are present in the F1 offspring of cannabis-exposed fathers. And our collaborators have observed behavioral as well as synaptic deficits in the offspring born to these fathers. And so with that, I will thank our entire team, and I will stop sharing my slides here. And yes, thank you.